Great, thank you. I'm glad to be here and looking forward to telling you a bit more about our potato breeding program. So we've got this sort of maybe intimidating title slightly, but hope to break it down over the next hour or so. We're going to talk about genomic selection, this methodology called optimum mate allocation, and applying this in both diploid and tetraploids, right? Potato that we grow commercially in this country are all autotetraploids. So I spent a lot of my career at UW trying to develop, extend the genomic selection framework to deal with polyploidy. So three different stories for you today. The first one is explaining what we call the directional dominance model. How do you account for dominance and inbreeding depression in an outbred crop, whether that's a diploid or in my case a tetraploid, and then some results for what that looks like in our breeding program. It's great to see all the applications of genomic selection that we have here today. What you will find, if you haven't already, if you start to do this for a couple years and you really go into it, you're going to start to wonder, wait, what's happening with my genetic diversity? And so that's the point I reached a couple years ago after about 3 years of doing this to realize I've got to get more serious. And I studied how it was handled in animal breeding, and they use methods that I'll talk about, the one today called optimum mate allocation. And then lastly, the kind of new frontier for potato breeding is how to transition from a tetraploid germplasm base to a diploid. So I'll tell you a bit about our progress on that front. So to begin, it's a real honor that I get to be the steward of the potato breeding program, which goes back to the 1930s when the first program director was hired. I'm the fifth program director. You can see some of the important milestones along the way. For potato varieties, I think a good benchmark, if you have really hit a home run, is that you grow it on 10,000 acres. Okay, right? You have to always calibrate relative to what the total acreage of a crop is, whether that's a big or a small number. For maize, that's not an impressive number. For potato, that's a pretty impressive number. So we've had two of those home runs in the history of the program. One in 1960, after the first 30 years, that was the variety called Superior. Then after 30 more years, a variety called Snowden. Both of these are potato chip kind of varieties. So we're kind of due, if you just think about that, very short time series. We haven't, I don't think we've hit it, unfortunately, since in my time yet, but I'm hopeful that it may be just around the corner. So here are some pictures of some varieties that we have developed in the last decade. And part of the challenge, I think, for me and some of the other people we've heard today is how do you manage, actually it's a lot of small breeding programs. Red potatoes would have red skin, the potato chip varieties, which are round white, the russets, those are all separate breeding programs. You cross a red skin variety with a russet variety, you don't get anything that anyone wants to buy, right? So managing separate germplasm groups, and we don't have all of these tools developed to the same extent in all of these different product profiles. So what does traditional tetraploid potato breeding look like? For those of you who've studied plant breeding methods, the clonal crop breeding chapter is usually the simplest chapter because you basically create the genotype and then you just start propagating it. It's pretty simple. That's what this looks like. We have what I think are beautiful, perfect flowers in potato. We sexually hybridize them. We do have a fair amount of male sterility problems and even female flowering problems, right? For a long time, if you just select only on the economically important part of it, the tuber, you can easily have some of your best clones that will never even flower when you put them in your crossing block. So that's a continual source of disappointment. We have cytoplasmic male sterility that we don't have full understanding of. But if you do actually make the cross, you do get typically very good true seed, as we call it, botanical seed production. That's not really a limitation. Typical number you see here that for each of these product profiles, let's say for like my potato chip breeding program, we might have on average 40 families per year. So a family is an F1 population with perhaps on average 500 offspring, so about 20,000 different genotypes evaluated per breeding cycle. So those true seeds are sown in flats and then transplanted out at a small stage into small pots and under some kind of protected culture. Could be, in our case, it's a high tunnel. Some people use screen houses, but not in a field setting. You're growing out those seedlings typically, and then you can get anywhere from 0 to a couple tubers for each one of these small pots. And you typically then take one of those tubers and plant it out in the field the following year, and this begins now the decade-long process basically of clonal multiplication and evaluation. So starting in this table, we count typically based on number of field generations. So in that first field year, we've already reduced, say, from the 20,000 down to 15,000 because it could be as much as 25% of them didn't produce any tubers at all when you had them as seedlings. And the rest of them are evaluated as single plants. So we have the most genetically diverse field of potatoes right in the state of Wisconsin, except for the Frito-Lay breeding program, which is right next door. So from that initial group, you probably throw away 90 to 95,000. Some programs even throw away a higher percentage. It's very high selection intensity, even though you're only evaluating very highly heritable simple traits, like does the potato have the basic shape and appearance characteristics, the tuber, to be marketable? Does it even mature, or is it still like a green tree after 2 applications of the dried down herbicide? I mean, those are highly heritable traits, and you still have a hard time selecting more than 5 or 10% based on those characteristics. The second field year, you now have multiple plants per genotype, so that's what we call the clone selection stage. In my case, that's about 750 genotypes per product profile, and we use 8 plants for that evaluation. And then it goes on from there. We start to have both our seed nursery and multiple locations for yield trials in that third field generation. That's like what a preliminary yield trial would be for a grain program, I would say, and then advanced yield trials, et cetera. Okay, so how does genomic selection play into all this? When I started 10, 12 years ago, we were using a SNP array genotyping, and at that price point, we could only really afford to genotype the preliminary yield trial. So we were genotyping 100, 200 clones per year, something like that. Several years ago, like we've heard already before about the RAMseq, these targeted amplicon sequencing platforms have really revolutionized what you can do because they're so much cheaper. It's about 20% the price of what our array genotyping was, so I can do 5 times as many clones per year. So now I can genotype my clone selection trial on the order of, let's say, 750 or 1,000 clones per year. And we have a 4,000 targeted GBS panel, and we can then make genomic predictions that allow us to advance from the clone stage to the preliminary yield trial and getting closer towards that product and variety release. And that's an important use of genomic selection. But I hope you've learned in your classes or through your research already that the real impact of genomic selection is the parent selection, not the advancement towards the product release, in terms of what the genetic gain improvements are over time. So how do you pick your parents to start the next breeding cycle? That's where genomic selection has its biggest impact. And when it comes to thinking about that, we have to talk a little bit about the components of genetic variation, and especially as a polyploid crop breeder, that's something that we think about a lot. So, not all of the genetic variation you measure is heritable to the same degree. When we talk about additive values, that refers to the portion of the genetic variation that is transmitted with the highest degree of efficiency. And in a diploid population at Hardy-Weinberg equilibrium, you could say that the breeding value, 2 times the mean progeny value of an individual equals its additive value. That's why we focus on the additive value portion when we make our genomic predictions. The additive value of an individual is the sum of its additive effects of the genes. That's the connection between when I say a value, I'm talking about an individual. When I talk about effects, I'm talking about the genes themselves. And what is this additive effect? It's defined statistically based on a linear regression of your genotypic value, or your trait, you could say, against the dosage of your allele. So here's a picture of real data from potato, which shows on the vertical axis vine maturity, our plant maturity, and the x-axis is the SNP allele dosage for one particular SNP that tags a really important gene for maturity. It's a tetraploid, so we can have 0, 1, 2, 3, and in fact, you could theoretically have 4 copies of this, but as you can see, the maturity is going down with each additional copy of this early maturing allele. If you are 4 copies in a tetraploid, if you were homozygous, those plants don't even make it to our yield trials because they're so early they don't yield essentially. So we never even observe that homozygous condition for the early maturing state in our tetraploid breeding program. Anyways, this is just giving you a visual of what additivity means when you talk about additive effects. And so it follows from this that the additive value can be written as a linear function of the allele frequency within an individual. Okay, That's not something, it's not a concept that we use a lot, but within individual, or a diploid can have a frequency of 0, a half, or 1 for a biallelic locus, whether you're right homozygous versus heterozygous. And tetraploids can have also frequencies of 1/4 and 3/4 because they have these 4 possibilities. And so you can make these kinds of figures which plot the additive value versus frequency, and the point is that no matter what ploidy you're talking about, it's always a linear relationship. That's one of the common things. And the slope of that line has something to do with the effect, the strength of it on the phenotype. Well, now let's talk about the non-additive portion, right? That's really what a lot of my research has focused on, how to theoretically define these quantities and then apply them. There's different components of non-additive variation, right? Of course, epistasis is important, but for people who work on outbred crops, I would say dominance is arguably the easier of the two to tackle, and so that's what I'm going to focus my attention on here. It also has the benefit that a dominance model is the simplest kind of quantitative genetic model that can account for inbreeding depression, which is a really important characteristic of many of our outbred crops, including potato. As they become more homozygous, they get weaker and weaker, and you need dominance to be able to explain that phenomenon. Just like we have additive values and additive effects, we have dominance values and dominance effects. They're defined analogously. Just to put a little bit of mathematical notation on here, what is the dominance effect? Right, the additive effect was the kind of regression on allele dosage. How do you define dominance effect? It's also a regression kind of definition, but what you're regressing is the non-additive value on the diplotype dosage. Right, you have to count how many pairs of, let's say, if I call my two alleles 0 and 1, you have 0, 0, 0, 1, and 1, 1 are the three possible diplotypes for a biallelic. And what's the dosage of those? In diploids, there's only one possibility for any given individual. In tetraploids, you can have more than one dose of a given diplotype. So that's a tongue twister and something to a meditation if you're not used to thinking about polyploid genetics. But one of the interesting theoretical ideas that I've introduced is that of a dominance substitution effect. I'm sure you've all talked about additive substitution effects, which is the additive effect of one allele relative to the additive effect of another allele. You can make the same kind of idea, but it has a little bit different mathematical nuance. The dominance substitution effect is the heterozygous dominance effect relative to the mean of the homozygous dominance effects. That's what that equation looks like. It looks like a mid-parent heterosis calculation for anyone who see that, and that's the mathematical analog, but now instead of we're talking about parents, we're talking about combinations of genes, so diplotypes. And you can put in words, you would say it's the average phenotypic change per unit increase in heterozygosity. Geometrically, which might be more intuitive for some people, is to talk about that it's a quadratic function of allele frequency. If additive values are linear functions of allele frequency, dominance values are quadratic functions of allele frequency, right? The heterozygote is superior to either of the homozygotes when you have dominance, and depending upon whether you can plus or minus can be good or bad depending upon the context, but we're going to be showing all these figures as in positive values are desirable in this situation. The center of that inverted parabola depends upon what you assume for the population frequency. So here I'm showing you the example where the population level frequency was 1/4. But anyways, this maybe gives you something to think about in terms of how do you generalize dominance to polyploids. You can see you can plot diploid and tetraploids on the same kind of figure and have it make sense. So from this sort of gene-centric type of definition of effects, we can construct statistical models that describe how are the additive and dominance values distributed in our population, right? If we make the assumption that the additive effects are normally distributed, the dominance effects are normally distributed, this leads to multivariate normal distributions. This is the top line says the vector U of additive values of your population is multivariate normally distributed with a mean of 0 and some covariance, which is this genomic relationship matrix G times some variance component. Okay. The dominance values are also multivariate normal. They have some dominance relationship matrix given by those quadratic kind of figures I was showing you a moment ago. But the key point is that the mean is not 0. This is where the inbreeding depression comes into the picture. The mean of that distribution depends upon two quantities. One, the vector of the inbreeding coefficients. So the different individuals have different genomic inbreeding coefficients, what does that measure? That just measures basically how much homozygosity is present. Individuals with higher homozygosity have a higher genomic inbreeding coefficient. And then you also have this parameter B, which can be called baseline heterosis, could also be called inbreeding depression. Those are kind of the same idea, just reversed. So that's a measure of a trait that describes how sensitive is a particular trait to inbreeding depression. The mean value is going to become more negative if you have a higher inbreeding coefficient, or you have more baseline heterosis, more inbreeding depression for that trait. Both of those things reduce the trait value. You have more inbreeding depression either way. So in practice, what does this mean? You compute these inbreeding coefficients from the marker data. You fit that as a fixed effect covariate in the model, and then the linear regression, the mixed model tells you, okay, well, based on that covariate, you estimate that B parameter. That tells you how strong is the inbreeding depression for that particular trait. Well, what are those estimates with some real data? So here are 3 traits that are important for our potato chip breeding program. We, of course, care about yield, dry matter, the amount of starch in the potato tubers, and then the fry color. We make a lot of potato chips, and we measure how light they are. The convention is that people want lighter colored potato chips. So the first column is, and this is showing you the relative amount of the dominance variance compared to the additive variance. We can see that for yield, which tends to show the the dominance is most important for yield of these 3 traits, we're almost at half as much variance. So, I mean, dominance variance is definitely not negligible for our most important trait, yield. And if you look at the second column, this is the inbreeding depression estimate scaled by the square root of the additive standard deviation. So it's a pure number. These are both ratios. They have no units on them. And we have, right, an inbreeding depression that's over 7 times the additive standard deviation. If you were to and what does that mean? Basically, if you were to fully inbreed the tetraploids to pure homozygosity, that's how the yield would be reduced by 7 standard deviations of the additive variance, right? That's significant inbreeding depression. So, and the software to do this is this package called StageWise that I've developed over the last couple of years. So hopefully this gives you a kind of a feeling for the kinds of genomic prediction models that we're using in a outbred polyploid crop like potato. What I want to do now is pivot and talk a bit about how we can use those kinds of predictions and the marker effects themselves to design our crossing block. And we're going to use this method called optimum mate allocation. And the key insight is that if you just were to compute breeding values and do a naive truncation selection, take your candidates, rank them from the highest breeding value to the lowest breeding value, and draw a line, you will be out of business in 5 years. Okay, if you actually follow through on that prescription, you have to. And the reason is that all of your superior individuals would be very closely related. All of these blunt methods, whether you're using pedigrees or genomic relationship matrices, strongly lead to selection of closely related individuals that have shared superior characteristics. So again, it's really good for short-term gain, but for long-term gain, you have to do something else to maintain that genetic diversity because you deplete it too quickly. And in animal breeding, the way they formalize this is by defining an inbreeding rate and measuring an inbreeding rate. So let me formally introduce that to you. Inbreeding rate is the amount of new inbreeding that's generated per generation. And it is defined based on a probabilistic measure of identity by descent, classically. So the inbreeding coefficient, F, in generation T plus 1, so the progeny generation relative to the inbreeding coefficient of the parental generation has this relationship that the two genes are IBD in the offspring dependent upon whatever your inbreeding rate is, and then the complement of that times whatever the inbreeding coefficient was of the previous generation. Now, for many of you who work on inbred lines, you don't really think about inbreeding. I worked on small grains for a PhD. I never really thought about inbreeding as a problem before. Well, what does it mean if you work on an inbred line crop? It means take your set of parents, randomly mate them for one generation, and that's the population that we're talking about. Now they're fully outbred, and the question is, how related are they? What is their, take two genes at random in a locus, what's their probability of IBD? That is still a very relevant measure even for an inbred line type of crop. This concept of inbreeding rate has a close relationship to something that I think in plant breeding we talk about more often, which is an effective population size. So the classical relationship is shown here, that the effective population size N sub E is 1 over the ploidy divided by the inbreeding rate. So animal breeders for decades have recommended you should be targeting an inbreeding rate of about 0.5 to 1%. That's, again, the probability of new IBD per generation, and that works out to, for a diploid, an inbreeding, an effective population size of between 50 and 100, which I would guess is something that seems reasonable to a plant breeder. For tetraploids, those numbers are half that number. So anyways, hopefully this is not seeming too foreign to you. Here's a simulation just to show the consequences of ignoring this important issue. This is done with the AlphaSimR program. The left is showing you the change in the genetic variance of the simulation over time. So at cycle 0, at the beginning of the simulation, we started a 0 change in genetic variance. And then over time, the genetic variance is going down. At the same time, on the right-hand side of the figure, the genetic gain is going up, right? You have to deplete genetic variance to make genetic progress. You have to deplete genetic variance to make genetic progress. That's like the fundamental theorem, basically, of plant breeding. The question is, how efficiently do you convert that loss variance into gain? That efficiency concept is really central to what we do. And you can see the 3 different inbreeding rates I've simulated. The red is 0.5%, blue is 1%, and green is 5%. Well, you can see we can rule out the green idea pretty immediately. It's losing genetic variance the most rapidly on the left-hand side. And, after 20 cycles of selection, it's got less genetic gain in the green than the other solution. So there's no benefit, right? You get less gain and you lose more variance. It's the least efficient approach. The 0.5 and the 1% are more of a toss-up. It depends upon your time horizon. I mean, if your job depended upon reaching a certain target in 10 years, you might say, well, I'm going to target 1% because, after 10 cycles, you do have a little bit of advantage of the blue compared to the red. But look, by 20 cycles, you basically, the 0.5% is caught up to the 1%. And now the 0.5% is about to overtake it. You can see the trajectories, and that's because less genetic variance has been consumed under the 0.5% inbreeding rate scenario, right? So you can't say for sure which is the best depending upon your time horizon, but somewhere in that 0.5 to 1% is a good rule of thumb to be thinking about as a breeder. Okay, so how can we do this in practice? How can we design a crossing block that targets 1% inbreeding, for example? The way that this was done traditionally in animal breeding was by a method called optimum contribution selection, or OCS, which was an optimization technique that determined essentially what contribution did any one of the parents make to the next generation, right? What proportion of the offspring can trace back to a particular parent, with these 2 goals in mind. They tried to maximize the average breeding value of that average. It's like a weighted average of all the parents. But that weighted average has to respect whatever the maximum inbreeding rate you specify. That was the idea. It's a great idea. I have used it for a number of years. The point of the OMA, the optimum mate allocation, seeks to answer the question, well, what if you go beyond random mating? This methodology of using breeding values assumes that you basically will take your parents and you'll randomly mate them. We know that certain mate combinations can exploit their specific combining ability due to their complementarity. Can you use that to your advantage? Instead of just randomly mating your parents, make specific crosses. So the F1 progeny mean is the sum of the mid-parent additive value. That's capturing that breeding value portion. And then we have this mid-parent dominance and this mid-parent heterosis. And so we're wondering, can we leverage those other parts of the genetic variance? So there's a useful acronym that we can talk about, which is genomic prediction of mate performance. Can you predict the average performance of a particular mating? This has also been called genomic prediction of cross performance, or GPCP. I'm just using the GPMP because it's more agnostic in terms of animals. They don't talk about crossing, they talk about mating. So it's a little bit more general, but it's the same idea. And if you are working with an inbred line crop, when you're making, we're basically talking about predicting hybrid performance instead of in this case, instead of there being a genetically uniform hybrid, we have segregation in that hybrid generation, but we can say what's the mean of the population. That's what we're talking about here. And one of the things that just published recently was a way to make these predictions for outbred crops, including polyploid outbred crops. And the solution turns out to have these kind of nice geometric interpretations in terms of what you would call like conic sections from a geometry. You have, if you're trying to predict the mid-parent dominance, the two axes are the allele frequencies in the two parents. So if they were contrasting alleles, if this was homozygous for one allele and the other parent was homozygous, you'd be looking at the two corners of this figure, but we can have intermediate values when you have outbred polyploids. So anyways, the mid-parent dominance are these circular curves of constant dominance value. The heterosis actually has elliptical level curves, and you put them together and you get these hyperbolic level curves. So some interesting theory there, which I'm not going to spend a lot of time on, but needless to say, there's a software now that can make these predictions based on just the marker. You have the marker effects that you estimate, and then you have the marker data for your parents, and it uses these formulas to make the hybrid predictions even when your parents are tetraploid potato. The software that does this I've called COMA, which stands for convex OMA, or convex optimum mate allocation. Instead of trying to optimize the contribution of every parent, it optimizes the contribution of every possible mating, which means you have kind of exploded the number of unknowns, right? If you have n parents, maybe you have a selection pool of 300 possible parents you're trying to pick. Well, there's on the order of 300 squared divided by 2, which is a lot more possible matings, and that's the number of decision variables that this software can solve. It uses very efficient optimization methods that go by the name of convex optimization. So because of that, it's the first time that we have a software that can guarantee optimality of the OMA problem. It offers the explicit inbreeding control as opposed to more nebulous penalties on diversity, which are useful but don't allow you to directly dial in, I want 0.5% inbreeding, for example. And of course, it works for polyploids. So let me show you some results from a simulation of applying this software to see how it performs. This is not a very complicated simulation. I'm just going to do a mass selection where I have a constant population size of 500 individuals that are phenotyped and then genotyped as well. I use that data plus the data from their parents and the grandparents, and so I have basically these 3 generations in my training population. You could do more, it just makes the size of the simulation bigger and take longer, and you can still see the effect with this level. This to make this interesting, we simulated a trait with dominance, right? Otherwise, it's not a very interesting simulation for these kinds of problems. You can see in this table for the diploid and the tetraploid simulations, there is substantial inbreeding depression and dominance variance, which is not exactly matching the real potato data I have, but it's in the ballpark. It's suggestive that this is going to be interesting. So here is the result from using COMA to manage genetic diversity and design the crossing block over 25 cycles of genomic selection. COMA actually, the software COMA can run in 2 modes. It will run the classical OCS version, where you don't look at specific matings, just the parent selection, and it runs this mate allocation version. And so the top panel here is showing you the inbreeding coefficient over time. So I'm showing you at 1% inbreeding rate and 0.5% inbreeding rate, with a 1% inbreeding rate starting at an average inbreeding coefficient of 0.1. It goes up to 0.3 over 25 cycles, and it goes up to basically only 0.2 if you're at 0.5% inbreeding rate. It's a little bit easier to see, does it actually work as promised? At the bottom, this is plotting the realized inbreeding rate right? You specify an inbreeding rate, then you can measure, did you achieve that? And you can see it's bouncing around the target. The 1% simulation is bouncing around 1%, and the same for 0.5%. So it achieves the desired inbreeding rate. This is based on, how do you measure inbreeding? I talked about probability of IBD. That comes from a pedigree model. So even though we're doing genomic selection to predict the merit and the marker effects, we are using the pedigree kinship to calculate our inbreeding rate. And a natural question is, well, why not use genomic relationships to calculate inbreeding rate, genomic inbreeding? The interesting thing is that genomic inbreeding is not based on probabilities. This is not something that is widely appreciated, but it's actually based on gametic correlations, you could say, at diploids. And the upshot is that you can't feed a genomic kinship matrix into this kind of software and get a sensible result. It has to have a probabilistic interpretation. Genomic kinships can range from positive and negative values. And so they're not probabilities. And so basically you can't use genomic kinship for coma and all the other typical type of OCS software that is available. It doesn't really control the inbreeding rate long term. Okay, how did the OMA compare with the OCS, right? The mate allocation with the parental selection, how did it compare in terms of the complexity of the mating design? That's what this attempts to show. The left side is showing the number of parents. So this is diploid, this is tetraploid. We've got our two inbreeding rates here, and here's the number of parents, right? So from the kind of calculation we just talked about a moment ago, what's the effective population size? If you are at a 0.5% inbreeding rate, it's around 100 for a diploid, and it's more like 50 for a tetraploid. And that's kind of similar to the numbers that we observe. And there's no real difference. The gray is the OCS and the white is the OMA. No real difference in terms of number of parents. But look at how different the number of matings required is, right? To do a proper OCS, you have to randomly mate your parents. And that can require hundreds of matings. Now, if you have a crop that is nicely wind pollinated and you just plant it in a field and it open pollinates, well, that's not a problem. If you're hand pollinating, like in potato, and making all those crosses, that's a significant logistical constraint. We can achieve the same or better genetic gains with a fraction of the matings, 100 matings versus 500 matings, and it gets even, the discrepancy is even greater in tetraploids compared to diploids. So significant logistical benefits to using this OMA method compared to OCS. And then of course the real test is, okay, what did you observe in terms of long-term gain and efficiency? And the gains are modest, I will say, in this perspective, but they are real. So this is a plot of genetic gain versus 25 generations, and the solid lines are the diploids. The solid line is the OMA. The dashed line is the OCS. The two colors are tetraploid versus diploid. So you can see the solid line is consistently above the dashed line, meaning we're getting slightly more gain using this optimized mate allocation compared to just a breeding value type of approach. Even when the gains ultimately kind of line up in the diploids by 25 generations, you can't say there's any benefit, but you look out into the genetic variance profile and you can see, oh, actually there's a little bit more genetic variance left after 25 cycles with the optimized mate allocation approach. So it's still more efficient even though the gains are pretty comparable. So easier to implement, better, as good or better performance. This is currently the workhorse of how we're designing the crossing blocks in the potato breeding program at Wisconsin now. All right, I'm going to, in my last bit of time, want to tell you a little bit about kind of the new frontier, moving away from tetraploidy and outbred based breeding designs to think about an inbred hybrid breeding system. Can we actually achieve that in potato? To do so, we have to first get to a diploid level because inbreeding is very slow. Besides the fact that there's inbreeding depression, it just takes a long time to actually achieve high rates of homozygosity when you have a polyploid. The long-term goal would be to release our varieties as true seed, right? Instead of having to release them as in vitro plantlets or as seed tubers. There's a lot of excitement in the private sector. New companies are being formed every couple of years to try and pursue this. Companies see for once now an actual return on investment potentially for potato compared to the traditional breeding schemes. We'll see if that all plays out, but basically most of the serious potato breeding companies that had traditional breeding programs have now created a diploid hybrid breeding program as well. What are the potential advantages of this breeding system? Well, in general, we can say genetic change is more rapid at the diploid level compared to the tetraploid level in terms of how quickly can you change allele frequencies. Of course, once we have inbred lines, we'll be able to use that nice method of backcross breeding to be able to introgress traits without changing all the other characteristics of the parents of our hybrids. And of course, we know we have heterosis. I've just estimated heterosis for you in our tetraploid potato program, but we don't really have a very efficient way of systematically exploiting it. And the thought is that with inbred lines, you can explore hybrid combinations and heterosis much more efficiently. These advantages will lead to higher value seed in terms of the genetic resistance profile. Growers will be able to reduce their pesticide inputs. That's good from a sustainability perspective. Of course, that will very likely lead to more expensive seed, and how that all plays out remains to be seen, but that should stimulate further investment in breeding for this new methodology. Another key point that's not so obvious to a breeder is that it's actually likely to be cheaper to produce the early generation seed for potato with a true seed kind of variety compared to a typical clonal variety, and that's going to lead to a shorter seed supply chain. I wanted to show you what that looks like because it's something that I think is getting the growers excited about this. This is what a typical seed supply chain looks like right now in potato, right? The current varieties, or you make a new variety, it gets released as an in vitro plantlet. And it takes 1 year, they multiply that in vitro, then they can put them in some kind of hydroponic or aeroponic system to harvest mini tubers. That takes a year. Those small mini or micro tubers are then planted with specialized kinds of equipment, very shallow in the first field year to produce now a typical type of field seed tuber. Those are multiplied for 2, 3, 4 more years, even before it ever gets into what we call a commercial grower. In the potato jargon, you have the seed growers who multiply the seed, and they are certified. They're constantly trying to worry about the levels of the virus and the more phytosanitary characteristics of their seed. And then you have the commercial grower, who's the one who has the contract to go to the grocery store or the fryer or whatever it might be, right? And they never replant their own material because it has, at this point, too much accumulated virus to be healthy seed for that again. But you're looking at a 5-year, it's 5 years from the time a grower says, oh, I think I might want to try that variety. If there's not already seed in the pipeline, wait 5 years, and then you now have seed actually to produce it at the typical scale. That's a pretty long horizon. What if you have true potato seed, right, the botanical seed? The economic projections are that you can, in that first field year, because we're going to shift and use a transplant-based production system, the efficiency of what modern greenhouse systems can do with transplants, and having essentially in that first year, instead of having a greenhouse system, you now have a combination greenhouse field system. You can produce field year 1 seed at potentially half or even a third of the price of what it costs right now to do it by mini tubers. It basically allows you to skip a year, and so a commercial grower could buy Generation 3 seed for the same price that they're currently buying Generation 4 seed. So even if the varieties don't have any new genetic characteristics to incentivize their adoption, just from the price of the seed perspective, that could drive the adoption of a new variety which is available as true seed compared to an in vitro plantlet. But of course, we expect that these new genetic characteristics are going to allow people to spend more on their seed because they're saving money potentially on their production, on their management costs. And so we might even shorten that seed supply chain down to only 2 years instead of 3 years. So dramatically potentially reducing the time to market of new varieties. I've been fortunate to be the project director of an SCRI project that is wrapped up now that we called the Potato 2.0 project, right? A little bit of a tongue-in-cheek in terms of going from 4 to 2 sets of chromosomes. Here are some of the key things we've done over the last 5 years, right? We have to first establish the diploid genetic base for all of this breeding. So we did haploid induction on our elite tetraploids. And for potato, our haploids have still 2 sets of chromosomes. They're diploids, right? They're not monoploids like what you're used to if you work with a diploid crop. So we've sequenced all of their genomes, and these plants are now publicly available. We're getting interest from breeding companies who want to use them for jumpstarting their breeding programs. We've been doing our own breeding, of course, in the public sector. Probably about 5 years in my program, maybe more like a decade, even before this grant started at Michigan State University. And they have already found some diploids, not inbred hybrids. These are just outbred diploids that are yielding as well as their tetraploid checks. Now, they don't have all the other characteristics needed, so it's not like they're about to release the first diploid variety. But a lot of the perception of diploid breeding was that you'll never match the yield of a tetraploid variety. That has been the kind of the negative stereotype, which I think has prevented investment earlier. And I think that they're dispelling some of these notions through their work. And we have to learn how to grow potato transplants, and not a horticultural practice that we use, really. So we're learning how to do that practically, and we bought our first vegetable transplanter a couple years ago. And how do you manage? What's the weed management system going to look like? The other components of a field management program. So those are all the things that we're starting to learn, and we're hoping to learn in some future grant proposals. All right. Kind of circling back to my bread and butter, what is genomic selection going to look like? I'm not going to diagram for you a complete idea of what a diploid breeding program looks like, because honestly, I'm still playing with ideas myself. But I have wondered the question, as we start to, we do have a somewhat regular diploid breeding program now. Can we leverage our historical tetraploid data, which is much more extensive in terms of the training population size and the number of environments in the field we've measured? Can we use that to predict our diploids? So what we're going to be doing is trying to combine our existing theoretical frameworks that have been published in other contexts. So there's already work done to ask, when you have a pedigree-based breeding value prediction, how do you combine diploids and tetraploids? So that problem was solved in 2018 by Hamilton and Kerr. And the animal breeders have also spent a lot of time thinking about multi-breed genomic prediction. If you have a training model for one breed of cattle, can you apply it to a different breed of cattle? And so they had already published some nice work about these type of multi-population genomic prediction models. So both of these things really fit our situation quite well. Basically, what you have is these kinds of block-structured genomic relationship matrices. If I have two groups, groups 1 and 2, in this case I'm talking about a diploid group and a tetraploid group, but it could be anything. You have the genomic relationships within group 1, within group 2, and then between groups 1 and 2, right? Those are the off-diagonal blocks. And each one of those parts of the matrix uses group-specific allele frequencies, and in my case, ploides now as well. You can take a look at what this means visually a little bit. So here's a PCA diagram when we calculated the normal genomic relationship matrix for about 2,000 clones. About a little bit over half are tetraploid, a little bit less than half are diploid. On the left is a typical genomic relationship matrix. Clearly there's population structure here. The diploids are coming from a different genetic background because we do already have a little bit of wild diploid influence in them. But if we use this type of structured formula, we can actually collapse that population structure down, and now we get a G matrix that's kind of better posed for doing genomic selection. It's interesting to look at genomic inbreeding coefficients. I was talking a moment ago about how they're not so useful for controlling inbreeding rate, but they are still useful in terms of thinking about signatures of selection, I would say. Here on the left is the distribution of the inbreeding coefficients for my diploids, and here it is for the tetraploids. This is 0 here, so not a lot of departures from Hardy-Weinberg equilibrium values as far as inbreeding goes in our tetraploids. They're very recalcitrant to change. In the diploids, you can very easily make change, and then we actually have a mixture of F1 and F2 individuals here. That's why we see this kind of heavy tail of some individuals which have very high inbreeding, relatively high inbreeding coefficients. But even so, the mode of the distribution is still negative, right? And I talked about how you can have negative values. Negative values means that you actually have more heterozygosity than Hardy-Weinberg equilibrium. And I think the implication is that even just in the process of generating these diploids, those that have excess heterozygosity are benefiting from some complementation of deleterious alleles, and that's why they are surviving long enough to even make it into our genotyping and field programs. There's tremendous genetic load in the potato genome. Okay, one little kind of mathematical idea for those of you who are interested in these kinds of models. How does the trait, so we've got this structure in terms of the two groups as far as their genotypes are concerned. What about the traits? Well, we invoke a correlated trait model. And the kind of weird thing about these models is that you have to formally define the cross combinations of a trait with a group, even though it makes no sense. So if I have, in this case, I'm talking about, let's say, traits A and B, and I have groups 1 and 2, you only really measure ever trait A in group 1 and trait B in group 2. There's no such thing really about trait A in group 2 and trait B in group 1. That's like saying, I'm going to measure diploid or tetraploid yield in a diploid potato. That's a nonsensical statement in English, but mathematically that quantity exists when you write these kinds of models, okay? We just don't look at those results, but they are there, and you need that to derive these kinds of covariance models. So the vector of the additive values has two parts. It has a trait covariance component, direct product with the genomic relationship component. And from the estimates of the genetic variances of the two traits and their covariances, you get genetic correlations. And that's really what we're interested in terms of understanding how well can I predict one group from the other. It depends upon that genetic correlation. We had 3 years where we had pretty much side-by-side experiments of our diploids and our tetraploids with about equal numbers in the two different trials. And without parsing this dramatically, these are plot-based heritabilities, which were similar, let's say, for these two traits, the yield and the specific gravity, which is our measure for dry matter. So really, heritability, in this case, even though the plot sizes are a little bit different, we can say heritability is not driving any real, can't really invoke that as an explanation when you see differences between some of these results. We compared the goodness of fit by the AIC, the Aikaike information criterion, for this 2 population model versus the 1 population model. Right, AIC, lower numbers mean a better fit to the data, even though you have more parameters. So these, negative 60 is a substantial drop. We are doing a better job of modeling our yield data by allowing for it to be a correlated trait instead of assuming it's the same yield in both diploids and tetraploids. The genetic architectures are different. And then you get out those genetic correlations. For yield, the genetic correlation was about 0.4 between the diploids and the tetraploids versus about 0.6 for specific gravity. So the kind of upshot here is that from these correlations, we can say that the cross-ploidy prediction is possible, but it's never going to be as good as direct selection, right? I mean, indirect selection has to have a higher heritability in order for it to be more efficient than the direct selection. And as I just shown you, we don't have higher heritability, at least at this level. And so, until we build up our diploid training model, there's some benefit, but we're going to be shifting towards a diploid genomic prediction model as soon as we have a comparable training population size. Okay, so some concluding thoughts. This first point actually maybe isn't even worth saying at this point. I sometimes speak to audiences that are quite skeptical about genomic selection. Doesn't appear to be the case here today. And I think part of the reservation is that people feel that they don't have a way of building a genomic prediction model for all the traits that they normally select on, and neither do I. I mean, I don't have genomic prediction models for all the traits I work on. I have it for the ones that typically show a fair amount of G by E or that are easy for me to measure. And the other traits that tend to be more maybe quality traits that I don't have very good high throughput screens for, I still incorporate in more or less formal ways. Sometimes it's more ad hoc, sometimes it's more rigorous. But I guess my advice is don't be waiting for the perfect ability to do genomic selection before you get started, which I think is not the case anyways. Hopefully I've convinced some of you that you need to get more formal about measuring genetic diversity through inbreeding rate, and maybe this software coma I've developed can help you with that. And although I'm really proud of what we've accomplished as far as genomics-enabled breeding for tetraploidy over the last decade, it's still tetraploids, okay? There's still a biological limitation to what you can accomplish compared to a diploid. And so we're also pretty bullish about what this future inbred hybrid breeding system is going to look like. I'll wrap up by thanking the wonderful group that I've got to work with. The work on the diploid breeding in particular was the work of a former PhD student, Lin Song, and also benefits from the expertise of Maria Carrazza-Harder, who's a scientist in my group. We had this Potato 2.0 project with, you can see all the different institutions there. We appreciate the funding from USDA NIFA for that and our other breeding projects, as well as the support from the potato commodity boards. And thanks for your attention.
So you had lots of great points about the optimal mate allocation, but just thinking on a practical level, when I think about most breeding programs, I mean, if you think about industry, they've been buying other programs, right? And I would migrate in some material. So you're mainly just talking about if you only keep your breeding program closed, but most people are going to migrate some stuff in, aren't they, over 25 years?
Yeah, it's an excellent question. How do you account for migration in these kinds of models? I don't have a good answer for that. I think that is the frontier that quantitative genetics and population genetics need to think about to balance it. Because you're right, this is based on a closed population kind of model.
Is there a way that you can account for compatibility? Sometimes like some clones are not good females or not good males, but in the context of the optimized mating model, can you, I mean, with all of the information we have now about genetics or so, can we incorporate those aspects related with compatibility, cross-compatibility? Thanks.
Yeah, I didn't go into a lot of the details, but in the tutorial that you can find on the GitHub site, it shows how there's actually, you can incorporate a lot of ad hoc constraints. If you only want some individuals to be considered as male versus female, I mean, the software works for dioecious species in general, so it has that. I go through an example in the tutorial about, I have my genomic predictions, let's say, for my quantitative traits, and let's say maybe I have another marker for a disease resistance trait that I don't formally have in my multi-trait selection index because I don't really know how to combine them. But I know that I want at least, every mating, I want at least one of the parents of a mating to have, a must-have resistance trait, for example. You can limit it will only consider matings that meet that criterion and other types. So it can incorporate a number of these practical logistical constraints. In practice, I'll say that I've never, in practice, realized the optimum solution from the software, right? I mean, because my plants, the ones that I thought were going to be best based on the model, again, maybe they don't flower because we're not collecting flowering data in our clone selection trial, which we probably should be doing based on these experiences. So what in the end, what happens often is that I typically see what crosses I did make, then we just count the seed, and then I run this optimization where I say that, okay, if I know I have to sow 100,000 seeds, the maximum contribution in any of a particular mating is limited by how many seeds I actually have on hand. So you can incorporate the actual inventory constraints on top of any genetic constraints with this software. It has a lot of these kinds of features to it.
Hey, great talk. A lot of it was beyond my statistical abilities, but I did have the same question as Seth did about the infusion of new germplasm along the way, but I want to take off on a slightly tangential topic related to that. So in that breeding program at Wisconsin, they for a long time were working with extraction of haploids and doing 4x, 2x crosses, and then back crossing the 4x derivatives of that to the tetraploids and creating true potato seeds for virus control and so on. And some of that I know was being implemented internationally, and I'm kind of curious whether or not you're using any of that tactic, and or do you know of others that are still using that kind of approach internationally?
Yeah, that's a great question, and nice to have your historical perspective, right? My predecessor, many times, I mentioned I'm the 5th director. So the 2nd director of the breeding program, Stan Peliquin, was a famous, great geneticist and also instrumental in trying to promote the use of tetraploid true potato seed varieties. You could potentially release a variety as a tetraploid true potato seed, even though it's not uniform. I mean, if you have inbred lines, the benefit is that the true potato seed is genetically homogeneous. But the potential benefits would have been that, in places that don't have developed seed systems where they can't really maintain vegetatively tubers, seed lots clean for long periods of time, there are these phytosanitary benefits, even if genetically maybe there are some disadvantages. What I have been told is that SIP doesn't do this anymore. In practice, there are no tetraploid TPS varieties that have been very successful. There is one breeding company. There was actually one variety released in the United States by a European breeding company maybe a decade ago. I believe that's no longer available on the market because the demand wasn't there. There's one Canadian breeding company, Tuberosum Technologies, which is still trying to do tetraploid TPS, but primarily, again, only for kind of South Asia or places which have very strong disease pressure. But I think in general, the heterogeneity of the tetraploid TPS varieties ended up being insurmountable, I think, is what happened historically.
We have time for one more short question. So if there's no more questions, please join me in thanking Dr. Endelman for his presence.